There's also a little bit of a personal history behind this. I usually don't talk about this, but mostly because I feel it's a little bit cheesy. But I ended up being a cancer patient early on in my 20s. There was a diagnostic process based on pathology, which was incorrect, and some biomarkers didn't fit that. And I was already in medical school. I was in my 20s, and I catch that, which I think ultimately had a big impact in outcomes. Was a decision like radiation therapy versus chemo plus surgery, and ended up being the second, luckily. But I think if I had gone to the wrong pathway, probably outcome would not have been as favorable.
Something that you just mentioned a short time ago, that possibly, comprehensive genomic, profiles can help with differential diagnosis, whether it is, making a call or deciding how to treat something that might be a cancer of unknown primary or something that is less clear pathologically.
This is Genetics for Healthcare, a podcast for patients.
This is Genetics for Healthcare, podcast for patients, where we bring experts on to talk around how we can bridge the gap between innovation in precision medicine and patient access. And it's a pleasure to have Dr. Carlo Bifulco, who is the Chief Medical Officer of Providence Genomics. Dr. Bifulco, I've followed you for quite a while in this space, and we've connected on different occasions, but it's really an honor and a pleasure to have you on our podcast.
Thank you, Ram. Honored to be here today, and thank you again for the invitation. Look forward to conversation.
Absolutely. So I understand you grew up in Italy, and you went to med school there. Kind of what was your background? What attracted you to, I guess, wanting to be a doctor and specifically being a pathologist?
Yeah, on the doctor side, I think was a desire to do science, but to do science which was impactful at the human kind of level. I think that's a very generic kind of thing that drives most of the people who enter the field of medicine. Pathology-wise, I think a little bit more of a complex history. But at a very high conceptual kind of level, I think is from my point of view, a stronger appreciation that biology drives diseases, and that if you want to change outcomes, you have to intervene on the biology. And I felt that pathology was among the many disciplines that you have in medicine, the one which was closest to the biology. There's also a little bit of a personal history behind this. I usually don't talk about this, but mostly because I feel it's a little bit cheesy, but also because I didn't want it to impact my career, but I think at this point I'm late enough. But I ended up being a cancer patient early on in my 20s. And it was a funny story. There was a diagnostic process based on pathology, which was incorrect, and some biomarkers didn't fit that. I was already in medical school. I was in my 20s, and I catch that. Which I think ultimately had a big impact in outcomes. Was a decision like radiation therapy versus chemo plus surgery, and ended up being the second, luckily. But I think if I had gone to the wrong pathway, probably outcome would not have been as favorable. So I spare a lot of details, but basically, I think that left me with a deeper appreciation of how important pathology can be clinically. And the other experience that I had was that at the time I was in medical school, later on, was the peak of the pandemic of HIV, and I had the opportunity of seeing a time where people were dying without us knowing what was causing the disease. It was still unclear that the virus was the agent behind this, and I was at the time in Milan, which was quite heavily hit by this for a variety of reasons. Large population, unfortunately, using Fox at the time. So you would have all these people in their 20s dying and no answers. And I went to I was basically doing research as a student, and I went through the opportunity of seeing Fox struggling with this, and ultimately coming to the conclusion that it was a virus, and starting to target that with therapies. And we will talk later about success stories in medicine, but I probably would make the case that HIV is one of the big success stories in medicine in terms of outcomes.
Yeah,
People,
absolutely.
yeah, yeah, people used to die. I suspect, I don't know the numbers exactly, but I think probably the median survival was probably around 6 months. And now I think people are impacted minimally in terms of survival thanks to antiretroviral drugs. So they're giving me like this strong appreciation of the importance of understanding biology and ultimately brought me into pathology.
That makes sense. That somewhere along the way, you made a conscious decision to become a forerunner in specifically molecular pathology. And your team, so I came across this article that was published in Journal of Clinical Oncology Practice. Obviously, you and your team published a number of papers throughout the year, but this one was particularly of interest to me, being in this field of precision medicine. As you know, that we've advanced, we had to have like flash frozen or fresh tissue in the past to get certain molecular answers. And we're still asking the same question, what patients are the most or least likely to benefit from certain therapies. So in this paper, widespread adoption of precision anti cancer therapies after implementation of pathologist directed comprehensive genomic profiling across a large US health system. Big title, we know, but you can look that up and find that on uh, JCO Practice. What was the thesis and kind of impetus for the study, and why was it important for your team at Providence to do this type of analysis of pathology led comprehensive genomic profiling for cancer?
Yeah, yeah. I mean, the fundamental concept is that cancer is a disease, a genetic disease, or a disease of the genome, right? And biology is very complex, but I do feel that there's strong evidence supporting that cancer is at least one of the main drive. Genomics is one of the main drivers of the disease. And if you believe that, and you go to first principles, you would reach the conclusion that testing the genome and understanding the genome is crucial for treating the cancers. And I think the logic of that is super stringent. The application of this in reality, though, has been extremely complex. And if you look at what happens, especially outside a few elite cancer centers, the reality is across the nation and I think across the world that most of the patients are tested suboptimally. They, at best get a few genetic tests, and those are super narrow. They select a few genes, the most common drivers of, cancer, and ultimately miss out therapeutic opportunities. And there are many reasons why that happens. I guess we will talk about those later on. But we wanted to remove all the obstacles, and I wanted to make sure that every patient had the opportunity to have comprehensive genomic profile of the tumor. And the way we decided to do this was to deeply involving the pathologists in the workflow. So we embedded the genomic testing into the diagnostic process. Um, I don't I don't know how familiar your listeners are with what happens in pathology, but typically uh, cancer is diagnosed after some kind of procedure. There could be a biopsy or resection, and people look at what comes out of that resection or biopsy at the microscope. Um, and that's the first time that there's certainty about a malignancy, and there's the first time that there's actually certainty about the type of malignancy. And what we decided to do was to link that process with genomics. Again, with the idea that genomics is ultimately the driver of the process and is what will give us therapeutic options. By doing so, we removed, all the headaches that arise and the delays and suboptimal tissue utilization, which arise with what happens currently most of the times in clinical practice.
You had a good call outs because many patients and their support team, whether it's family, friends, are not aware of the cancer diagnostic journey before they unfortunately experience it firsthand, right? As you mentioned, they will meet with a surgeon or some specialist to get a piece of tissue to be able to do the workup. From there, they may meet with a radiologist, another surgeon, medical oncology, and other providers during the workup, but they may not meet with a pathologist, but it's the pathologist who provides the answers for all these other specialties. And your department is involved from, I mean, the beginning where you're talking about screening and you just diagnostic workups to the end of the treatment journey and into survivorship. And that's why I felt this paper and the approach was so important, that pathology is the point, team in saying this patient needs, profiling, and to implement it in the diagnostic workup. Why don't you think more hospital pathology departments take on the mantle of leadership to order, or at least initiate the conversation around genomic or genetic testing?
Yeah, it's a great question. Rome, I wish I had a simple answer. I think the answer is, my question is, that there are multiple factors which make this difficult currently. Yeah, they go through, there's a big range of reasons. Some of them have to do with technology. The technology itself, I would argue, is not novel anymore. It's been around for quite a while, but it is, because of the breadth of the data that you're generating, is still something that is unfamiliar and a little bit challenging for many pathologists to departments to handle. And so that's one component. There is a reimbursement landscape, which is challenging. I guess we'll talk about it later. But basically, Medicare, the government, CMS, reimburses this kind of testing, but um, there's a variety of insurance providers which still consider this experimental, and that provides problem from a reimbursement perspective. Um, there is also a cultural thing with pathologists. If you think about pathology, the field has been, I would submit, a little bit static. The technology that we use is 150 years old, more or less.
Hmm.
Don't quote me on the precise number, but basically is a byproduct of German chemistry. The stains that we use are coming from German chemistry in the 18th century. The light microscope has been around for quite a while. While I think the field has made progress and is tightly integrated with clinical workflows and so forth, I think there's also a little bit of reluctance in adopting all technologies. So yeah, so it's a composite kind of picture. Another thing I want to mention is that pathologists themselves, as you pointed out, are often not visible to the patients, and I feel that often their perception is that their main relationship is with the clinicians, so with the
Hmm.
oncologists or the surgeons. So they see themselves as you know the helper of the surgeon or the oncologist, which is great. But I also feel that we have, I think our first mandate is to serve the patients, right? And I think that position of not serving the patient directly, but through another, to me, that somehow loses some muscles, which I think are important, actually.
Yeah.
So yeah, so a complex answer, and I apologize for that. But yeah, and I'm sure there's more that I missed, but those are a few things that keep to my mind.
Yeah. Do you think there is somewhere in the future a role for pathologists to, let's say, have like clinic hours for patients? Because they we have genetic counselors who are the subject matter experts for germline genetics, but we really don't have anyone in the healthcare system that is that designated go-to person for clinical genomics for treating cancer and soon to be other diseases. But what do you think about like pathologists possibly having office hours to meet with patients for this reason or any other specialty that is connected to the lab?
It's an interesting idea. It's possible. I have to be completely honest with you. I do think that medicine is going to change dramatically in the next 5 years because of the impact of a thing called AI. I think it will dramatically impact pathology, but it will impact everything, the whole stack. So I do think patients in the future are going to be very different from what they are now. They will be coached by AI early on. That's my
Hmm.
expectation. Conversations are going to be very different. Um, I struggle to tell you how things are going to be reconfigured in the future. If I've, in the not too far future, but I really think the medicine is going to be a different beast from what we see now. And to be honest with you, I do feel that that's going to be great for patients ultimately. The transition is going to be a little bit challenging, but I do think it's going to be a very different configuration.
Yeah. And the goal and the reason why we have these conversations on genetics for healthcare is patient access. We want to ensure that patients have information that can empower them to have these partnership conversations for shared decision making. And in that spirit, the results in the paper, which was really robust, by the way. So anybody listening to the podcast, I recommend that you go take a look. There will be a link on our page. But I just want to get into some quick study results, Dr. Bifulco. So a total of 3,216 patients with advanced cancer. 80% of those, over 80% of those were stage 4, were tested. And obviously, there's when you get into these numbers, there's always details behind the details. But at a top line, 49% of patients had at least one actionable mutation, meaning an FDA-approved drug for those that had comprehensive genomic testing, as opposed to smaller panels. 53% of the patients in the study were eligible for at least one of 3 large basket trials. And obviously that could have been more patients found eligible because if you were looking at the broader world of clinical trials, right? So you also compared in this study, which I thought was very interesting, patients who received comprehensive genomic testing, which is called CGP, comprehensive genomic profiling, and your product, your test that you integrated in house was testing 523 genes, and you compared it to a simulated model of patients who would have had only 50 genes. Now this is a I thought was pretty major. 67% of those patients who were tested with CGP were found to have actionable gene mutations for an FDA approved therapy or a clinical trial versus 33% of patients in the study with 50 genes. What you just called out, um, just in our conversation, and you also do this in the paper, is the significant variability, in testing clinically. And there's likely, because of many different factors, going to be variability, but what patients don't really benefit from is the unwanted variability, meaning are you testing CGP versus single gene versus a smaller panel? But what are some of the reasons for this variability in testing that you've seen?
Yeah, yeah. So in the real world, I think most of the patients, and even today, are unfortunately, at least at first line kind of setting, tested with sequential kind of genomic testing at best. So they will have a small panel. They will have some kind of um, um, fish kind of test done. And for folks who are unfamiliar, fish is fluorescent hybridization for detecting amplification of, for example, HER2, or for detecting diffusion, and often have multiple kind of events of this sort. Um, and that kind of pattern, I think, is the pattern which is currently built into the pathology workflows almost everywhere. And so, one of the reasons why it is adopted, I think, is a little bit of inertia. We've talked about before about the challenges of bringing CGP in a clinical kind of environment. And we talked a little bit about, why pathology hasn't been playing a very active role, or not as active as I would like to. Um, but, uh, um, but yeah, so all those things basically um, bring us to a situation where most of the patients are are currently tested with this kind of, in my opinion, suboptimal kind of pathways, and what that translates into is, as you mentioned before, less genes tested, which means less available therapies. I also want to point out that also it may translate, and it can translate for sure, also in the wrong diagnosis, unfortunately, because the genomic information is not only important in driving therapies, but also important in the diagnostic process. We could talk about that a little bit later. Um, um, and uh, um, and also, it results in delays in terms of availability of information. So one of the things that also we highlight in the study, I think, is how quickly were we able to give information to the oncologist, and um, and I think it was statistically very clear that by doing what we did, which was basically triggering the testing at the time of diagnosis by pathologists, we made sure that information was available to the oncologist the first time of the visit with the patient in a very significant number of cases, which is obviously not true with the other pattern. Um, so we benefits from multiple point of views, from a diagnostic point of view, from easiness of availability of information, and also comprehensiveness. One additional thing I want to mention is that very often when you do this kind of sequential testing that I described before, one gene at a time, one test at a time, you risk of losing all your diagnostic tissue. So every test consumes some of the tissue. And for some procedure and for certain diseases, particularly, think of a brain tumor. As you can imagine, people don't like to remove large chunks of brain, so the tissue will be often very limited. Or think about a lung cancer, which also typically is assessed by a transbronchial biopsy or thoracic biopsy. But as you can imagine, people don't like to take out like chunks of lung if they can avoid it, and very often, that kind of intervention is not therapeutically feasible. So tissue is minimally, it's super minute, very, very, very small. And if you think about every test consuming tissue, you often end up in a situation where you cannot do anything anymore because all the tissue is gone. There's also, an advantage because you eliminate that kind of scenario from the get-go, right? You get all the information that is currently important from the treatment perspective at your first chance, and that simplifies workflows and and can work very well. And I think that's what the paper describes. It can work very well in clinical practice.
And I guess a better way that I guess I could have said is the unwanted variability is test versus no test at all, which is not necessarily as helpful for patients. Now,
Yeah,
your approach at Providence, and I want to emphasize this, uh, this is no casual undertaking to integrate NGS testing. All of the factors that you just spoke about that can result in variability of test CGP versus a single gene versus a smaller panel. There's a lot of factors that go into how those decisions are made, as you just mentioned, right? There's the availability of tissue, the timing of when that decision is made, the comfort of the treating physician to interpret and apply those results that you don't know right before you get the results. So all those, there's just so many different things, and I'm not here to say that there's a right or wrong, but I want to focus on what Providence had to do to integrate this types of testing. Can you talk about the challenges of just simply integrating, NGS testing? You got sequencers, you got people, knowledge systems, and other tools. Like, how did you think about that to stand this up?
we were fortunate. First of all, we had leadership support in the organization for this, which I think was essential, right, for making it happen. The other thing I want to mention that over the years we have built a great team. One of my coordinators is Brian Pining, super active in the genomic space, and so we had a fortuitous opportunity to actually execute. Um, one of the strengths also that we had was the fact that all information is at Providence is in the cloud. So Providence has been, I think, one of the most active healthcare systems in the country in terms of moving to the cloud from an IT perspective. And I think that it has enormously facilitated a lot of our analysis and workflows in general. It also enabled our compute piece. I think, NGS, as I mentioned before, is relatively novel. I don't consider it novel anymore, but we have been with the technology from the get-go, from the beginning. We were one of the first groups to adopt this, and I can tell you the early days were quite challenging from a compute perspective. Um, and the technology would change continuously, and so the availability of having a cloud environment for doing computation and so forth really simplified our workflows, enabled us to go through multiple transitions over time, and has enabled us to be on the cutting edge, pretty much. The last piece I want to mention that for whatever reason, I'm probably one of the few pathologists who actually have computational kind of background. I like to write software. Believe it or not, I actually lead the bioinformatic team here, and that helps tremendously early on. I don't think that's a requirement anymore. Software vendors and the technology providers like Illumina and others have, provided now make available solutions which are almost closed box, put a tissue in, get results out. But over the years, that was certainly not true, and I think it was a huge advantage to be able to be agile in that kind of space and be able to move rapidly and quickly. So some challenges. I do think that the technology is getting more mature, and that many of these challenges are going away, um, and that the future enables you an easier kind of entry. Actually, I do think that most of the obstacles that I mentioned before are basically gone, or will be gone very soon.
Essentially, what we're talking about is like decentralization. And for those out there, um, the for this specialized testing, genetic testing, you have central labs, which are larger laboratories where hospitals will send tissue out to those labs so they can do the testing and get a result, and for many different reasons. And that's why this paper kind of shows that your thinking is ahead of the curve. And speaking of decentralized testing, AI brings hope that tumor diagnosis can be possibly more efficient, that tumor profiling can be decentralized and integrated into the workflow at diagnosis to integrate those genetic data points into the healthcare ecosystem, if you will. And you and others were recently quoted in an article in the MIT Technology Review talking about the challenges with AI that includes, when we're speaking about pathology, that only 10% of pathology practices in the US are digitized, and that we also need much larger training sets, which means like, the space and storage required for this is massive. But your team at Providence, along with Microsoft, have developed GigPath, which is an AI pathology model. Can you talk about how this might be able to remove some barriers for patient access to genetic genomic testing, and how it can, I guess, reduce some of the variability in testing that we just talked about?
I mean, we talked a little bit about the potential of AI to transform healthcare, and thank you. You're referring to some recent work that we had. We have a paper published in Nature with a Microsoft team and actually UW, a collaboration of three institutions. Hoi Fung Poon has been a great partner from an AI perspective for us, and the paper is focused on using AI to and specifically what we call a large foundation, transformer-based models. I'm sorry for the many words there, for actually driving the future of pathology. So super is a super exciting time from the perspective. If you follow the news, we're recording this today. I think I think a week ago you had DeepSeek R1 going live, a Chinese kind of model which performs AI work compatible to the best models of OpenAI in an open source kind of fashion going live. And that it just tells you that the AI is going to accelerate even farther. At least that's my impression. Yeah, so pathology will also be part of that, and as the MIT paper alludes, I think there's it's still early days though. What I want to mention is the fact that digital pathology, so the ability to actually digitize slides completely and not look at them in a microscope, but actually look at them on a computer screen, has been around for almost, I think 20 years, but the adoption has been very slow. It reminds me a little bit of the discussion that we just had about genomic testing and being only, slowly adopted in pathology. And I think we're still early steps in that kind of process, but there are incredible opportunities, and I really believe that the AI solutions that are going to become available will drive a much quicker adoption of digitalization and actually, I personally feel there is going to be a future where all the imaging modalities are going to coalesce in some level. So you're going to have radiology, pathology, all part of the same kind of integrated kind of platform, where you look at the body with an MRI, but at the same time the pathology and with microscope, but the two get integrated through the use of what we call multimodal kind of models. I think the future is going to be even more interesting because the multimodal models will bring in also the genomic information into the conversation. So it's going to be genomics, multiple different types of imaging tools, and the clinical data that are in the EMR all integrated in one single solution, and that the process that we work through will be mediated through AI. Uh, yeah, so a very interesting future, and I think some of the problems that we described in the MIT interview, which have to do with the fact that only a small portion of pathology is currently digitized in the US and in the world, I think are going to, those dynamics are going to change because of the opportunities which will be made available by AI itself. Um, so yeah, so very hopeful in a positive feedback loop there. Um, AI also is important for other things that we do. I didn't mention it, and it's not described in the paper, but we also have AI solutions which facilitate clinical trial matching. If you think about genomics results, we talked about the impact on patients in terms of first line therapies and second line therapies, FDA approved drugs. But there's also a layer where unfortunately patients fail multiple rounds of therapies, right? And then ultimately end up in a situation where the clinical trial is the only option. And I don't know how much people are aware of this, but unfortunately, only 5% of the eligible patients actually end up enrolled
Yeah.
in a clinical trial. So there's a huge gap there, and we have been using AI to try to close that gap, and we have AI solutions which go over clinicaltrials.gov. Parse that content, extract the crucial information which is needed for the matching, which typically is genomic kind of information, then use the AI tool to analyze the clinical pieces of information, the genomics as we mentioned before, and then offer opportunities to patients to enroll. So we feel that there is an ecosystem which is going to be supported in addition to the pathology piece, but also goes also into the electronic medical record. And hopefully in the future, as was mentioned before, multimodal, right? So also, imaging and other things will feed in a in a comprehensive solution. I also want
Yeah.
to point out this ecosystem kind of idea. I think it's great to do genomic testing, but you also want to make sure that people get treated and enroll in clinical trials as an outcome of that. And so there, my opinion is that you need to have an ecosystem to support genomics for clinical delivery, and that has also been a big focus for us.
Yeah. I think this is bolstered by the commitment of the current administration to invest up to 500 billion in the Stargate project, right? That is going to build and AI infrastructure. And so, AI could play a key role in healthcare sooner than we thought previously before. And obviously, the reason is to broaden treatment opportunities, as you just mentioned. And so, diving back into the paper, there's some key outcomes and data points that I thought were significant. So, twice as many patients received comprehensive genomic profiling. Twice as many patients received immunotherapy or targeted therapy, versus those who did not find a mutation because they had a smaller panel, right? So 13.6% doubled over 6.4%. Four times as many patients were eligible for immunotherapy, targeted therapy, or a clinical trial. The 50 gene test patient cohort who was found not eligible for any trial or any therapy, again, 67.4% of the time versus 33% for the more comprehensive. Patients with up to 6 months or more of follow up, 52% of those patients got targeted therapy versus 32% who got chemotherapy. It assumes that they had the smaller panel. But the patients who got targeted therapy also had significantly improved overall survival compared to patients who got chemo alone. What does this study say to providers and patients about tumor profiling for cancer care?
I think that's the gist of the message. Better outcomes, right? Better survivals, and I think it's a reflection of the genomic testament. It's also a reflection, I think, of a paradigm kind of shift in the treatment of patients. Um, we're moving from chemotherapy, surgery, radiation, things that are fundamentally killing cells or removing them to more sophisticated kind of approaches, which are really driven by biology. And In a way, we talked about HIV at the very beginning, but I think this is another manifestation of the progress that you have in medicine and really a paradigm shift in the way we treat cancer patients. The numbers are significant, and also at the same time they hide some pretty transformational things. I just want to mention, for example, melanoma, which was disease untreatable 15 years ago, and now a lot of patients have long-term survival. So when I go to meetings and I give talks, I all the time meet with patients advocates who are long-term survivors of of lung cancer, melanoma. They're 5 years out. They're not the average patient, but those things were impossible 15 years ago. So it's a reflection of a changing landscape. And again, I think this hopefully is only the beginning of what is possible. This is the current state of the art, but I do think that in the future, with the use of AI, early interventions, liquid biopsy, which we will not talk today, I imagine, because of reason of times, but MRD, mean residual disease, we neoadjuvant therapies, immuno-oncology, and so forth. There's much more that we can do, and I think we have a bright future from a medicine perspective. Um, I almost feel that we will look back to the past as the dark ages of medicine, and um, I will have the opportunity to see that day.
It kind of echoes the sentiments of Dan Von Hoff, who is a friend, very much a forerunner and a thought leader in the area of precision medicine. He says you can only find it if you look for it, which is 20 years ago he was advocating for comprehensive genomic workups, and we didn't really have NGS as a commercial technology then. But even he talked about biomarkers like tumor mutational burden, right? And in this paper, that was the most frequent biomarker detected across all main cancer types. So you can't assess that with smaller or single genes. and that can unlock access to therapy. And I think that something that you just mentioned a short time ago, that possibly comprehensive genomic profiles can help with differential diagnosis, whether it is making a call or deciding how to treat something that might be a cancer of unknown primary or something that is less clear pathologically.
That's a super important point from my perspective, it's a little bit of a source of frustration for me in the pathology community. Some components of pathology are moving forward more rapidly. So if you think about neuropathology, they're incorporating genomic information in the diagnostic name of the entities they diagnose. So genomics becomes a crucial piece of giving a name to a tumor. I think the same is true for hematopathology. For when you talk about malignancies like leukemias or lymphomas, the genomic information is an essential piece of the name that gets assigned to the disease. Somehow that has not happened yet for lung cancer, colon cancer, and so forth. Some folks have argued that should happen, and they have my support. Let's put it like that.
Well, I always ask, especially when we get the opportunity to have, great thinkers, researchers like yourself, for patients and their family, their care support team, what are three questions that they could ask their doctors or their providers to help advocate for a comprehensive genomic, profile approach to their cancer care?
One question which comes to me, but maybe because I'm biology driven, would be what is driving my tumor, and if you have that conversation, ultimately you end up into genomic testing. That is the very first one. The other thing that comes to my mind is what are the consequences for my family of this diagnosis? It's often overlooked, and we didn't talk much about germline testing, but and it was not really part of this paper, but one of the things that happens when you do this kind of testing, you also find germline alterations, and those have potential impact for relatives, right? Because they could have a cancer predisposing mutation, and I think the conventional number is around 10% of a cancer patient having those kind of germline alterations, and they can be incredibly impactful for the family members, because we have ways now to screen them early, identify the tumor early as an approach, or even having interventions like surgeries to remove the risk of having a tumor in the future. So those are the first two. The third one, and we talked about that late before, has to do with clinical trials. I do feel that the best therapy for patients is a clinical trial, almost always, and that applies in situations where you have failed therapies, but also potentially in conventional therapies, but also potentially in situations where you have not failed on therapies, but there are other options which are available. And so asking about clinical trials, I think, should be part of the conversation, even at an early stage. So what drives my tumor? What are the consequences for family members? And lastly, clinical trials are three things that come to my mind. I'm sure there are others equally important. Yeah.
Well, we'll certainly take that from you, Dr. Bifulco. Dr. Carlo Bifulco, CMO of Providence Genomics. Now, I mean, you're publishing, you're integrating, and these are not easy things to lead, because you need a massive team to do that. You're developing AI tools. My goodness, do you have downtime? What do you do to relax?
Good question. I actually I really like the outdoors. I like to kiteboard. I just came back from a short break kiteboarding in Baja California for a few days. So though work is a big driver, I have ways of relaxing and and getting in touch with reality at a different kind of level, which I think is very important.
Well, you live in beautiful Portland, so it's easy to do that in the Northwest. Again, Dr. Bifulco, thank you so much for being a guest on Genetics for Healthcare podcast for patients.
Thank you, Rome. It was great talking with you, and thank you for your insightful questions.